Hello everyone, in this lecture I will explain the design and working of an EMG controlled prosthetic hand used in rehabilitation engineering. This technology helps amputee patient regain hand movement using muscle electrical signals. There are three commonly measured bioelectrical signals in biomedical engineering. The first is electroencephalography or EEG, which records the electrical activity of the brain. The second is electrocardiography or ECG, which records the electrical activity of the heart. The third is electromyography or EMG, which records the electrical activity produced by skeletal muscles. Among these signals, EMG signals are very important for prosthetic control. When a person attempts to move a muscle, electrical signals are generated in that muscle. These EMG signals can be detected using electrodes and used to control prosthetic devices such as artificial hands. Therefore, EMG plays a major role in rehabilitation engineering applications. So next, block diagram of biosignal acquisition system. This diagram shows the basic bioelectrical signal acquisition and processing system used in biomedical engineering. The human body generates different type of electrical signals as a result of physiological activities. These signals can be measured using specialized electrodes placed on different parts of the body. For example, EEG electrodes are used to measure electrical signals generated by the brain. ECG electrodes measure the electrical activity of the heart. EMG electrodes measures the electrical signals produced by muscle contractions. These signals are initially analog signals and cannot be directly processed by digital system. Therefore, the signals are passed through an analog to digital converter, also known as ADC, which converts the analog signals into digital form. After conversion, the signals are processed using digital signal processing techniques. DSP helps in filtering noise, extraction features and analyzing the signals. Finally, the processed data is sent to a computer where it can be visualized or stored or used for further application. In rehabilitation engineering, EMG signals obtained from muscles are commonly used to control prosthetic device such as artificial hands. So next we are going to see generation and decomposition of EMG signals. This diagram explains how EMG signals are generated and processed. When the brain sends a command to move a muscle, the signal travels through motor neurons from the spinal cord to the muscle fibers. These motor neurons are called alpha motor neurons and they control contraction of muscle fibers. When motor neurons activate the muscle fibers, electrical signals known as motor unit action potential are generated. A motor unit consists of a motor neurons and the muscle fiber it controls. Electrodes placed on the surface of the skin detects these electrical signals produced by the muscles. The signal recorded by the electrode is called the raw EMG signal. The raw EMG signal is complex combination of signals generated by multiple motor units in the muscle. Therefore, signal processing techniques are used to separate or decompose this complex signal. This process is called EMG signal decomposition, where the raw EMG signal is divided into individual motor unit action potential trains. By analyzing these motor unit signals, we can understand the muscle activity and use the information to control prosthetic device such as artificial hands. Next we are going to see two methods for acquiring EMG signals. First one, intramuscular electrode, second one, surface electrode. Now we will see in detail. So intramuscular electrodes are the electrode that are inserted directly into the muscle tissue. These electrodes are usually thin needle electrode or fine wire electrode. Because they are placed inside the muscle, they can record electrical signals from a very small number of muscle fibers. This allows researchers and clinicians to obtain highly accurate and detailed EMG signals. Intramuscular electrodes are commonly used in medical diagnosis, neuromuscular research and clinical studies. However, since they are invasive and require insertion into the muscle, they are not continuously used in everyday prosthetic control system. So next one is surface electrode. Surface electrodes are placed directly on the surface of the skin above the muscle. These electrodes detect electrical activity generated by muscle contraction through the skin. Unlike intramuscular electrodes, surface electrodes are non-invasive and do not require insertion into the body. They are easy to use, comfortable for patients and widely used in rehabilitation engineering application. However, surface electrode records signals from a large group of muscle fibers, so the signals may contain more noise compared to intramuscular recordings. In most prosthetic hand control system, surface EMG electrodes are commonly used because they are safe, convenient and suitable for everyday use. But intramuscular electrodes are not suitable for everyday use. So next we are going to see some applications of EMG. So we already know EMG is a technique used to measure the electrical activity produced by the skeletal muscles during contraction. One major application of EMG is in clinical and biomedical diagnosis, where it helps doctors detect neuromuscular disorders and nerve damage. EMG signals are also widely used in rehabilitation engineering, especially for controlling prosthetic devices and robotic exoskeletons. These systems allow amputee patients to control artificial limbs using their muscle signals. Another application of EMG is studying the recruitment order of motor units, which helps researchers understand how muscles are activated during movement. Finally, EMG can measure the activation level muscles, which is useful in physiotherapy, sports science and rehabilitation. Therefore, EMG plays an important role in both medical diagnosis and advanced assistive technologies. So next slide we are going to see data acquisition from EMG signals. The first step in prosthetic control is EMG signal acquisition. EMG stands for electromyography, which measures electrical activity generated by muscles. In this system, we use silver chloride electrodes. These electrodes can be placed either on the surface of the skin or directly inside the muscle. When a person tries to move their hand, muscle generate electrical signals. These signals are captured and sent to the processing system. So this figure shows the block diagram of the EMG based prosthetic hand control system. First, EMG signals are collected from the muscles. Then the signals are filtered to remove noise and unwanted interference. After filtering, feature extraction is performed. In this work, autoregressive model coefficients are used as features. These features represent the characteristics of the EMG signal. Next, classification algorithm identifies the intended hand movement. Finally, control signals are sent to the prosthetic hand to perform the required motion. So in this slide, EMG data was collected from 5 amputee subjects. Sensors were placed on the muscle responsible for finger movement. 3 finger movements were analyzed, that are index finger, middle finger and ring finger. Each finger movement was recorded under 3 speed conditions, that is slow, normal and fast. This helps the system understand different movement patterns. This slide shows the EMG signal pattern recorded for a specific finger movement. The horizontal axis represents time and vertical axis represents the signal amplitude. We can observe different signal characteristics depending on the speed of movement. These variations help the classification algorithm identify the intended motion of the user. This slide shows the EMG signal response during slow movement of the ring finger. When the ring finger moves slowly, the muscle contraction occurs gradually and produces relatively low electrical activity. As a result, the EMG signal amplitude is lower compared to faster movements. These slow movements help researchers study fine motor control and precise muscle activation patterns. In prosthetic hand systems, identifying these slow EMG signals helps the controller detect gentle finger movement. Next, EMG signal response during normal speed movement of the ring finger. During normal movement, muscle contraction is stronger than slow movement but not as intense as fast movement. Therefore, the EMG signal amplitude is moderate. This type of signal pattern represents the normal hand movement we are performing in daily activities such as grasping objects or typing. So, the EMG signal response during fast movement of the finger. When the finger moves quickly, the muscle contract rapidly and produce stronger electrical signals. As a result, the EMG signal amplitude increases significantly. The waveform becomes more intense and shows rapid fluctuations. Detecting these strong EMG signals allows the prosthetic control system to recognize faster and more forceful movement of the user. So in this slide, the EMG signal response during slow movement of the index finger. When the index finger moves slowly, the muscle contraction occurs gradually. As a result, the electrical activity generated by the muscle is relatively small. Therefore, the EMG signal amplitude appears lower compared to faster movements. These slow movements help researchers understand fine motor control and precise muscle activation. So next we are going to see index finger fast response. When the finger moves rapidly, the muscle contract more strongly and quickly. As a result, the EMG signal amplitude increases significantly. The waveform appears more intense and contains higher variation in signal strength. These strong EMG signal help the prosthetic system detect rapid user intention and respond quickly. Next we are going to see index finger response for normal speed. During normal finger movement, the muscle contract with moderate strength. Therefore, the EMG signal amplitude becomes moderate compared to slow movement. This type of movement is commonly observed during daily activities such as pressing buttons, typing on a keyboard, or picking up small objects. So by studying these EMG patterns helps the prosthetic control system identify natural hand movements of the user. So in this slide we are going to see thumb finger slow, fast and normal speed movements. So first we are going to start with thumb finger response during slow speed. The thumb plays a very important role in hand function because it helps in gripping and holding objects. During slow thumb movement, the muscle contraction is gentle and produces relatively small electrical signals. Therefore, the EMG signal amplitude appears lower. Detecting this signal is important for enabling fine and precise movement in prosthetic hands. The EMG signal response during fast movement of the thumb. When a thumb moves quickly, the muscle contract more rapidly and generate stronger electrical signals. This results in a higher EMG signal amplitude. This signal indicates strong muscle activation and rapid movement intention. By detecting this signal, the prosthetic control system can execute faster and stronger gripping actions. Next we are going to see thumb response for normal speed. During normal thumb movement, the muscle contract with moderate strength. As a result, the EMG signal amplitude becomes moderate. These signals represent typical daily hand activities such as holding object, writing or gripping the tools. Understanding these EMG patterns helps the prosthetic hand perform natural grasping activities. So next finally we are going to see conclusion. EMG based prosthetic hand systems provide an effective solution for amputee rehabilitation by capturing muscle electrical signals and processing them using signal processing techniques. It is possible to identify the user intended movement. The prosthetic hand then performs the desired action. Further research focuses on using artificial intelligence and machine learning to improve accuracy and real-time control. I hope you all are understand the concept. Thank you so much for watching this video. Thank you once again.